Yes, okay, so today is the 16th. And this is one where they're talking about sex linkage. They're talking about something that is an XXXY chromosome sex determination. That's the pattern that we see in humans. There are other species where there are different patterns, but this is the pattern we find in humans. Which pattern of inheritance would support the hypothesis that a trait being studied is a recessive sex-linked trait found on the X chromosome? We didn't really spend a lot of time with sex linkage, but remember that females get 2 copies, males in general get 1. So if a female has a bum copy of an X, she has a backup. So generally she's not affected by the trait. If a male gets that bum copy, he's got no backup. So he will be affected by the trait. So the first thing is anything that's recessive X-linked is going to primarily affect males. So right now, mostly expressed in females. We can get rid of that. Expressed in half the females. Well, no, that would be outrageous. So now we got to limit it down to the next 2. Trait is only expressed in males who have a father with the trait. So let's say that you have a biological father, which all of you do. If you are a girl, and I'll draw a little girl symbol there. I'll move that up. You got an X from your mom. What'd you get from your dad? An X. Yeah. If you got an X from your mom and you got a Y from your dad, you're a boy. So we would not have an X-linked recessive being passed from father to son. So that's bogus. Trait is mostly expressed in males who have a maternal grandfather with the link. That's the right answer. But we haven't talked about this enough for me to feel like you just get it. So I'm going to tell you a brief story. This is actually about my husband, who is colorblind. So let's go to a clean sheet. The other thing that we're going to talk about at the end of the day are pedigrees. We're going to wrap up with a little talk about pedigrees. But I'm going to do a pedigree here to kind of show you. There's my husband, and there's his brother. Now, in a pedigree, we color individual. Boys are squares, girls are circles. I don't know why. Somebody decided that. We color affected individuals as a solid shape. So there's my husband. There's his brother. His brother is not colorblind. And my husband, we know, is X, defective X, colorblind X, Y. His brother got the normal copy. Now, there's my mother-in-law and my father-in-law. So where did my husband get this X? If he got an X from his dad, he'd be a girl. He got it from his mom. They both got their Y from their dad. So we know that his mom, who is not colorblind, has to be X big N, X little n. Now, where'd she get that? So she is one of 5 kids. There were 3 girls and there are 2 boys. And of course they had biological parents just like everybody else. So, where did my mother-in-law get the X little n? Well, there are 2 possibilities. She could, so possibility number 1 is that her mom might have been a carrier, might have had that little n and she passed it on. We would assume to about half her daughters. Statistically, it would be about half the girls would get that. The other possibility is that their dad was colorblind. And he passed on that small X then to all the girls. We don't know. Her parents are both dead. I mean, she's in her 80s. You know, we can't, she doesn't know that if her dad was colorblind or not. But when we see a trait where maternal grandfathers have it, and then their grandsons have it, we would tend to think, yeah, that's probably X-linked recessive. Questions on that one? Okay, so this one is covered in the gene linkage video. And I said, there are traits that seem to go together. Redheads and freckles. Oh, I got three freckled redheads in here. So, doesn't mean that there aren't people with dark hair who have freckles. But I don't think I've ever met a redhead who didn't have freckles. Those traits seem to go together. And there are other traits that we see in, like in cats. What is it? White cats are often deaf. And there used to be an old saying among veterinarians, white feet don't treat, because there is a mutation that makes dogs sensitive to several kinds of common medications. And something like 90% of the dogs who have that mutation have like a significant amount of white fur. So those two traits seem to travel together. Well, this is the crossing over thing. This is how we get traits that either get mixed up or travel together. What's the result of this process? I normally do make a chromosome in 30 minutes or less with you guys. We didn't get a chance to do that this year. This is how you get grandma's eyes and grandpa's nose. This is how we mix up genetic material. So what's the result of this process, this crossing over? Is it an increase in variation? Well, yeah, that sounds pretty good, but let's look at all the other answers. The deletion of amino acids in a protein. When would we have that happen? Would that happen here, or would that happen in protein synthesis? That would potentially happen in protein synthesis. So that's bogus. Increase in mutations. Are we copying DNA right now? No. We're just making eggs and sperm. So that's out. The production of gametes that are genetically identical. Would this give you identical offspring or identical eggs and sperm? No. We're mixing stuff up. We're spreading variation around. So this one is, in fact, A. Question. Okay. Do you remember Punnett squares from middle school? When I did the little Punnett square review. But you've never done a dihybrid Punnett square. When there's a Punnett square on the air test, it's going to be a dihybrid. But in the last few years, when there were, and like I said, 2 years ago there were none, they've given you the gamete setup. So they've already told you what combinations of genes the eggs and sperm are capable of carrying. Here, all we have to do is figure out how many yellow, black, and chocolate labs we've got. And what they tell you is that if the gene, the eumelanin gene, is EE, big E, or big E, little e, then big B, and big B, little b give you a black lab, and little b gives you a chocolate lab. But if that EE gene is both copies of a recessive, then anything is going to give you a yellow lab. So it's kind of like an override switch. And you have to figure that out from the text. We haven't, this is, it's not epigenetics, epistatic genes. So polygenic is where multiple genes control a single trait. Eye color is polygenic. This is epistatic, where you've got one trait that's actually controlled by a couple different genes. So, again, 15 years of teaching biology, bachelor's degree in biology, I would write that down on my piece of paper because I can't juggle that in my head. It's too much. So especially when I do genetics problems, I write down the phenotypes, write down the genotypes, write down the genotypes and the phenotypes that they would create. So what I know right off is that any animal that's going to be little e will be yellow, and I don't have to do any more work. So where would I have, so I have a little e there, and a little e there, and a little e there, and a little e there. So I know that these animals would be little e, little e. So I can write yellow there, and I don't have to think about anything else on those 4. All right, so now I'm down to who gets a dominant gene, who's a black lab, who's a chocolate lab. Well, I've got a big B here, and a big B here, and a big B here, and a big B here, which means that every single lab is going to have one big B. So what color are all the rest of the labs? Do we have any chocolates in here? Not a one. These are all, it's going to be a litter of black and yellow labs. It is impossible for these 2 parents to produce any chocolate labs. Now, it doesn't mean that they're going to have 16 pups in a litter. It doesn't mean that they're going to have 4 yellow and 12 black. But what it does mean is that they absolutely cannot produce, will not produce, any chocolate labs. So these are all going to be black. How many of you figured that one out successfully? That one was really tough, and especially because we haven't spent time on epistatic genes. So I found that one challenging. Again, when you take the test, you're going to get scrap paper, so make sure you're writing stuff down. Okay, if there's anything that I love more than dog genetics, and I do think dog genetics is really cool, it's chicken genetics. I have a deep affection for chickens. Don't ask. I suspect that this is actually about a breed of... Who has chickens? You've got chickens. Anybody else have chickens? Okay, so in my dreams, when I hit the lottery, which I don't play, and I run my ranch for old, elderly, geriatric dogs who nobody wants, I'm also going to have heirloom chicken varieties, and Javas are one obsession of mine. Can you see this with the greenhouse and the old dogs laying around and the chickens? And I'll bake scones, and people will come just hang out with old dogs. It'll be wonderful. You're welcome to come. But anyway, in my little fantasy world, Java chickens have a black phase, a white phase, and it's called blue. It's like a speckled phase. So here we've got chickens, and it says that they think that this feather trait is incomplete dominance. So again, this is one more thing that we haven't really talked about, but I'm going to just do some brief work with you here. Incomplete dominance is when neither trait's dominant. And if we remember back to the stone crab allele thing, where big B was one color, big B, little b was a separate color, and little b was a third color, that's incomplete dominance. So here, what we'd be looking at is that big B gives you a black chicken, and big B, little b gives you a blue chicken, and little b gives you a white chicken. So the student hypothesizes this, and then they go out and they let chickens make babies. So which one of the results that they've got here would support their hypothesis? So I'm going to write that down anytime I have a genetics problem. Write down your genotypes, write down your phenotypes. So what if I had a black chicken and a white chicken, big B, crossed with little b, and I got blue chickens? Would that support my hypothesis? It would. Is there anything else that would support my hypothesis? So this is that scenario. So if I had white feathers and black feathers, and I got white feathers, no, that would not support my hypothesis. What if I had white feathers and white feathers, and I got black feathers? Nope, that would not support my hypothesis. And what if I had blue and white, and I got blue out of it? All blue out of it? Well, no, that wouldn't work either. So, I do these little sort of test crosses. So obviously this is A. Do you have questions on these? This is the world's briefest overview on pedigrees. Pedigrees are just ways of tracing genetic relationships and family relationships. And we can trace any trait. We can trace freckles. We can trace crooked pinky fingers. We can trace hanging earlobes. We can trace hemophilia. We can trace cystic fibrosis. We can trace sickle cell anemia. We can trace color blindness. We can trace anything we want using a pedigree. So in a pedigree, like I said, boys are squares. Whoops. Boys are squares, girls are circles, and affected individuals are colored in. So here we've got two unaffected parents. That line between them is a mating line. It's sometimes called a marriage line, but frankly, we don't care about marital status. And, you know, chickens, goats, and other things don't get legally married, so we'll just call it a mating line. It means they made babies. So if we have, wow, that's a really squished male. There. Okay. In this scenario, we have two unaffected parents who have a daughter who's affected. They have two girls and a boy. Daughter is affected. If I tell you that this is a recessive trait, and we don't know what the trait is, and we honestly don't care, can you figure out anybody in this picture's genotype? So their phenotype is affected or not affected. Can you figure out the genes they have? So big R, big R, big R, little r, and little r, little r are your possible genotypes. And this is a recessive condition, yes? Ooh, which both? The parents?
Yeah.
I agree. How do you know that?
Because the kids can, or one of them have the phenotype and the other two won't.
So you actually made the logical leap from, you figured out her phenotype, the affected daughter, and you went backwards and figured out the parents. That's some pretty high level thinking, bud. So she has to be little r, right? She's affected. Well, if she's affected, that means both her parents had to be carriers. There's no possible way for two unaffected individuals to have a child who is affected unless they're both carriers. Do we know her siblings' genotypes? Nope, we don't. It's possible that they're carriers. It's possible that they did not inherit that gene. So we sometimes would write that as R question mark. We know that each of them have to have at least one of the dominant unaffected alleles because neither of them are affected. How would we know? What would be the thing that would tell us if one of her siblings was a carrier or not? If they had a child and one of their children was affected. Now, I'm going to do, this is a weird little thing. I'll see if you can figure this out. So this is the symbol for identical twins. When the two lines come off, or I'm sorry, identical twins, I got that wrong. Identical twins come down and then off. So what do we know about this other twin? Two things we know. Seth, give me one of them. The male or female?
They're male.
It's got to be male because they're identical twins. What else do we know? Hannah?
One of them has to be. Would the other one be like affected?
They have to be affected, yeah. This is genetic because they share 100% of their genes. So if one's affected, we know the other one's affected. Now we know about her brother. We know he has to be a carrier, and so does his wife. We still don't know about this sister, and if she never reproduces, we might never know. Questions? That's a really brief, like I said, some years there are no pedigrees on the test, some years there are, but the thing that they always want you to do is use them. You know, figure out how to figure out genotypes, from them, how to know if it's, and they always use recessive conditions. So with that, I will stop the video.